Hello, my name is Brandon Montez, and I am a student researcher at Southeastern University. And specifically, I am involved in the efavirenz team, in which we focus in a single pause synthesis and just a more efficient synthesis of this drug that is currently established. And today I'll briefly be going over sort of the biology of reverse transcriptase and how it's involved in HIV, and in theory, how efavirenz actually inhibits or has been shown to inhibit the function of this enzyme, thus limiting viral replication. So in figure one, we kind of see the actual structure or representation of the structure of reverse transcriptase. We have the P66 region, which is also known as the palm domain, and the P51 region, which is also known as the thumb region. So the palm region, or P66, is actually the catalytic region of this enzyme. And as highlighted in the blue over here, it is actually where efavirenz actually binds to this enzyme and exerts its function. Now, exactly how efavirenz sort of limits the function of reverse transcriptase is in its hydrophobic interactions once it reaches that little niche pocket in the enzyme. And what essentially happens is that through these hydrophobic interactions, you get a 40-degree rotation between the palm region and the thumb region, or P66 and P51, and then the viral DNA is no longer able to carry out, be replicated, and DNA polymerization is essentially halted whenever efavirenz binds to this part of the enzyme. And in figure two is kind of a more cartoon version of what efavirenz actually does. So as a non-nucleoside reverse transcriptase inhibitor, it enters sort of the pocket in P66 and thus inhibits DNA polymerization once it fits into the reverse transcriptase and sort of um, results in that 40-degree shift within the actual enzyme in itself, preventing its proper function. And so we see how this drug sensitive virus, through the introduction of efavirenz, we sort of get that inhibition of DNA polymerization and thus viral replication. But this is how reverse transcriptase is involved in HIV, and how our drug efavirenz actually inhibits the function of the enzyme and is basically it's found to be crucial in its inhibition.